In this video, we'll be looking at the content within the four topics of this paper: cell biology, organization, infection and response, and finally, bioenergetics. In this episode, we'll start by looking at some key differences between prokaryotic and eukaryotic cells. Then look at types of organisms which fall into these two categories, examine typical cell structures of animal and plant cells, and finally look at bacterial cell structure. So, prokaryotic cells are smaller than eukaryotic cells, with an average size of around 1 micron depending on the bacterial strain. Animal eukaryotic cells are larger at around 25 microns on average, and plant eukaryotic cells can be even larger at approximately 50 microns. These are averages and will depend on the specific organism and cell type. Another key difference is that the vast majority of eukaryotes are multicellular. A multicellular organism is an organism in which many cells are working together to make up the organism, whereas prokaryotes, such as bacteria, are classed as microorganisms. A single cell is the entire organism. Now let's go over which types of organisms fit into these two cell types. Prokaryotes include bacteria and archaea. Archaea are a more primitive type of bacteria found in extreme environments such as thermal vents, glaciers, and salt lakes, whereas eukaryotic cells make up animals, plants, protists, such as parasites, and fungi, such as mushrooms. Next, let's talk about their subcellular structures and their functions, starting with animal cells. Key subcellular components found within a typical animal cell include a nucleus, ribosomes, cell membrane, cytoplasm, and mitochondria. Let's dive in and learn more about the functions of each of these. The nucleus controls what the cell does. It contains the DNA, which gives instructions for which things to make, and therefore which characteristics to express. Next, we have the ribosomes. These are very small spherical organelles which can be found free or bound to other structures inside the cytoplasm. It's the site at which proteins are made after taking instructions from DNA via the mRNA. And then we have the cell membrane. This is a barrier which controls what enters and leaves the cell. It readily allows vital small molecules such as water, glucose, amino acids, and gases such as oxygen and carbon dioxide to pass through via diffusion. The cytoplasm is the substance within a cell in which all the subcellular structures reside, and it is also where all the cellular reactions take place. So, what's powering all of the reactions inside of a cell? It's the mitochondria, also popularly known as the powerhouse of the cell. This generates energy for all the reactions going on inside the cell. But not all eukaryotic organisms have the same cell structure. In addition to all the cellular components found in animal cells, plant cells also have a cell wall, a vacuole, and chloroplasts. The cell wall is a layer surrounding the cell membrane and provides support and structural integrity to plant cells. The vacuole is a cell sap cavity within the plant cell used for storage of water and nutrients such as minerals and sugars. Whereas animal cells get glucose from eating, plants generate their own glucose through a process called photosynthesis. Now let's move on to bacteria, a prokaryotic organism which shares many of the components found in eukaryotic cells, such as a cell wall, a cell membrane, cytoplasm, DNA unbound by a nucleus, and ribosomes. However, it has additional features such as plasmids and a flagellum. As mentioned earlier, bacteria are unicellular microorganisms. Therefore, they evolved with a cell wall that acts almost like a shield to protect them from their environment. Additionally, bacteria have small circular pieces of DNA called plasmids. These provide a convenient, easy way to transfer useful genetic traits between different bacteria. Finally, Bacterial cells also have a subcellular structure called the flagellum. This looks almost like a tail and gives them the ability to swim and navigate through their surroundings. In this episode, we'll explore what cell differentiation and specialization is, at which stage differentiation occurs in different organisms, examine examples of specialized cells, and finally, we'll look at how these different cell types work together in a multicellular organism. Not all cells look the same. In fact, cells in your body have many different structures to suit their different functions. This variety of cell types is essential for complex organisms like humans. Without specialized cells, we couldn't have all the different tissues and organs that make up our bodies. Let's explore how cells become specialized through a process called differentiation. Differentiation is the process by which a cell changes to become specialized for its particular job. As cells differentiate, they develop different subcellular structures, and these structural changes enable them to carry out specific functions. When does differentiation occur? For most organisms, it happens during development. In most animal cells, the ability to differentiate is lost at an early stage. After they become specialized, they can't change into a different type of cell. However, plant cells are different. This is why plants can often regenerate entire structures from just a small cutting. In mature animals, differentiated cells are mainly used for repairing and replacing cells. This includes cells like skin cells that are constantly being shed and replaced. Blood cells also need regular replacement as they wear out. Some cells in our body remain as undifferentiated cells. These are called stem cells. We'll learn more about them in a future episode. Now let's look at some specific examples of specialized cells and their functions. Sperm cells are tailored for reproduction, functioning to deliver male genetic material to the female egg. With a long tail for propulsion and an abundance of mitochondria for energy, they are well suited to travel to the egg. Enzymes in the head region enable the sperm to penetrate the membrane of the egg. Neurons or nerve cells are adapted for fast signal transmission. They serve to relay electrical impulses across various parts of the body. Their elongated structure helps them span considerable distances. Branched ends enable them to connect and form intricate networks with other neurons. This network is what allows your brain to communicate with the rest of your body. Next, we have muscle cells. These are designed to contract rapidly. Their length provides ample room for this action. High mitochondrial content fuels their contraction process. Without specialized muscle cells, we wouldn't be able to move any part of our body. Plant cells also have specialized cell types, such as root hair cells. On the surface of plant roots, root hair cells develop thin extensions that protrude into the soil. These extensions greatly increase surface area, aiding absorption of water and minerals. These cells use active transport to take in minerals from the soil. Plants also have specialized transport cells called phloem and xylem. These cells align to form tubes for transporting substances across the plant. Phloem moves sugars and nutrients from leaves to other parts of the plant. Xylem, in contrast, carries water and dissolved minerals upward from the roots. These transport tubes are formed by cells connected in long chains. Xylem consists of thick walled, hollow cells, and phloem has minimal internal structures. This structure facilitates the smooth flow of materials through the plant. Cell differentiation and specialization are fundamental to multicellular life. They allow different parts of an organism to perform different functions. This division of labor makes complex organisms like us possible. Our next episode takes a look at microscopes to understand how we came to know so much about cell structure. In this episode, We'll start by looking at a diagram of a microscope, then take a look at the different types of microscopes and learn their key differences, explore an important equation that allows us to calculate magnification, and finally look at what standard form is and why we use it in science. Scientists first came to see individual cells with the help of light microscopes. The main components of a light microscope include the eyepiece through which a person looks through, objective lenses which are involved in refracting the light to magnify the image, a light source which amplifies the light entering the objective lens, a stage which holds the sample or specimen, a coarse focus adjuster for big shifts in focus, and a fine focus adjuster for smaller shifts in focus, and finally, an arm and a base for the microscope. Microscopes work by showing large images of very small samples or specimens. They magnify small objects with the help of light and lenses. However, the ability to see smaller subcellular structures such as ribosomes and plasmids was not possible with light microscopes. This was made possible with the help of more powerful and complex microscopes, known as electron microscopes, which were developed in the modern age. Whereas light microscopes use light to magnify an image, electron microscopes use electrons. These electron microscopes provide greater magnification and resolution. Resolution is the ability of microscopes to distinguish between two points next to each other. You will also need to understand how to calculate magnification. Magnification is calculated by dividing the image size by actual sample size. Additionally, you can also calculate any of the other components from this equation if you have the values for the other two components. Remember to make sure the units you use for the values are consistent. Convert any before calculating if required. As you can see, magnification is a very big number. Science has a way of dealing with very big or very small numbers, and no, it's not via the use of force. In science, we use something called standard form. This simplifies and expresses a long sequence of numbers in a very short form by shifting the decimal place. The first number in standard form is a number between 1 and 10, followed by a decimal place, followed by one or two other numbers found in the original value. This is then followed by a multiplication by 10 to the power of either a positive number if it's a very large number or a negative number if it's a very small number. The power of the number dictates the number of times the decimal point moves left for small numbers or right for large numbers. In this video, we will look at the process of binary fission, bacterial growth using mean division time, how microorganisms are cultured in laboratories, and finally, we'll see how scientists test the effectiveness of antibiotics. So, what exactly is binary fission? Well, the name gives us a clue. Binary means two, and fission means splitting. So, binary fission occurs when a single bacterium divides, creating two genetically identical daughter cells. But there's more to this process than just simple splitting. Let's break it down step by step. First, the circular DNA and any plasmids within the bacterial cell replicate. As this happens, the cell grows larger to accommodate the newly duplicated genetic material. The replicated circular DNA strands then move to opposite ends or poles of the cell. Next, the cytoplasm begins to divide and new cell walls start forming. Finally, the cytoplasm completely divides, resulting in two daughter cells. Each daughter cell has one copy of the circular DNA, though they can have different numbers of plasmid copies. What's truly remarkable about binary fission is the speed at which it can occur. When environmental conditions are optimal with sufficient nutritional resources and appropriate temperature ranges, certain bacterial species, including E. coli, can undergo a complete cell division cycle in approximately 20 minutes. Just imagine 100 bacterial cells could become 200 in 20 minutes, then 400 in 40 minutes, then 800 in an hour. This is why bacterial infections can develop so rapidly in the body. However, if conditions become unfavorable, perhaps due to lack of nutrients, extreme temperatures, or the presence of antibiotics, bacteria will slow or stop dividing and may eventually die. Because bacteria multiply at an exponential rate, scientists use mathematical formulas to predict population size. If you know the mean division time of a bacterial cell, you can calculate how many cells will be present after a certain period. Let me show you how this works with an example. Say we have a bacterial cell with a mean division time of 45 minutes, and we want to know how many cells it will produce after 3 hours. First, we convert all our time measurements to the same units. 3 hours are switched to 180 minutes. Next, we divide the total time by the mean division time to find the number of divisions. 180 minutes divided by 45 minutes equals 4 divisions. Finally, we calculate 2 raised to the power of the number of divisions, so 2 to the power of 4 equals 16 cells. So after 3 hours, our single bacterial cell would have multiplied to become 16 cells. Now, let's move on to how scientists grow bacteria in laboratory settings. This process is called culturing, and it's essential for studying bacterial characteristics and testing antibiotic effectiveness. To culture bacteria, scientists need to provide them with all the nutrients they require. This is done using a culture medium, a substance containing carbohydrates, minerals, proteins, and vitamins necessary for bacterial growth. Scientists typically use two distinct types of growth media, either liquid solutions called nutrient broths or solidified media containing agar. Agar is particularly useful because bacteria grown on it form visible colonies that scientists can observe and count. First, hot liquid agar is poured into shallow plastic dishes called petri dishes and allowed to cool and solidify. Once the agar has set, bacteria can be transferred to it using sterilized tools like inoculating loops. These are small wire loops that can pick up tiny amounts of bacteria. Alternatively, sterile droppers and spreaders can be used to create an even layer of bacteria across the plate. For safety purposes in educational settings, microorganism cultures are maintained at temperatures of 25 degrees Celsius or below, since dangerous pathogens tend to proliferate more readily at higher temperatures. However, in industrial settings where safety measures are more robust, cultures might be incubated at higher temperatures to accelerate growth. One of the most important applications of bacterial culturing is testing the effectiveness of antibiotics. This helps scientists discover new antibiotics and determine in which ones work best against specific bacterial strains. Here's how this investigation typically works. First, an agar plate is prepared with an even covering of bacteria spread across its surface. Next, small paper discs are soaked in different antibiotics or different concentrations of the same antibiotic. These discs are placed on the agar surface, spaced apart from each other. As the antibiotics diffuse into the agar, they will kill bacteria that are sensitive to them. After incubating the plate for about 48 hours at 25 degrees Celsius, clear zones called inhibition zones appear around the discs where bacteria have been killed. The diameter of these clear inhibition areas relates directly to antibiotic potency. More effective antibiotics produce larger zones where bacterial growth is prevented. A control disk soaked in sterile water rather than antibiotic is always included. This ensures that any observed inhibition is due to the antibiotic itself and not some other factor. When working with microorganisms, preventing contamination is crucial. Unwanted microorganisms can ruin experiments and potentially cause harm. Here are three key steps scientists take to maintain uncontaminated cultures. First, they sterilize inoculating loops by passing them through a flame before transferring bacteria. Additionally, they will seal petri dishes with tape to prevent airborne microorganisms from entering. And finally, they will store these petri dishes upside down to prevent potentially contaminated condensation from dripping onto the bacteria. These precautions ensure that the only microorganisms growing in the culture are the ones being studied. Binary fission and bacterial culturing are fundamental concepts in microbiology with widespread applications in medicine, research, and industry. Understanding these processes helps scientists combat disease and advance our knowledge of these microscopic organisms. First, let's go over exactly what we'll cover in this video. We'll begin by exploring what chromosomes actually are and how they store your genetic blueprint. Next, we'll look at the big picture of cell cycle and how cells prepare for division by growing and copying their DNA in preparation for mitosis. And finally, we'll take a look at the process of mitosis as one cell becomes two. Let's start with the stars of our show, chromosomes. These incredible structures reside in the command center of your cells, the nucleus. Think of chromosomes as the library books of life. Each one contains thousands of chapters called genes. These genes hold instructions for building and maintaining your body. So, what are chromosomes made of? They're molecules of DNA that have been super condensed and packaged. When you hear people talk about your DNA, they're talking about the information stored in these structures called chromosomes. Here's something cool. Your body cells have two copies of each chromosome. One set came from your mother, and one set came from your father. That's why you might have your mother's hair color but your father's height. In humans, we have 23 pairs of chromosomes, making a total of 46 chromosomes in all of our cells except in gametes, which we'll cover in a later episode. That's your genetic blueprint, the complete set of instructions for making you, you. Now let's talk about cell cycle, the life story of a cell from birth to division. Your body is made up of trillions of cells, but you started as just one cell, a fertilized egg. How did one cell become trillions? Through the process of cell cycle and mitosis. The cell cycle allows for three important things: growth, replacement, and repair. It helps you get bigger as you develop, it swaps out old and damaged cells with fresh new ones, and it heals wounds by creating new cells. The most dramatic part of the cell cycle is mitosis, the actual division of one cell into two identical cells. And each new cell created through mitosis contains the exact same genetic information as the parent cell. Before a cell can divide, it needs to prepare, just like you might prepare for a big trip. This preparation happens in two main stages: growth and DNA replication. First, the cell grows larger. It also makes more cellular machinery like ribosomes and mitochondria, the power plants of the cell. The cell essentially doubles its content, so it has enough to share between two future cells. Second comes one of the most amazing processes in biology: DNA replication. During this process, the cell makes a complete copy of all its DNA. Remember those 46 chromosomes? Every single one gets duplicated. The result is two identical copies of the original DNA. These aren't actually new chromosomes. Each X is really one chromosome that's been copied. At this point, the cell has copied all its genetic material and is ready for the main event: mitosis. Let's break it down step by step. First, those X-shaped chromosomes line up perfectly at the center of the cell. This alignment is crucial for making sure each new cell gets exactly the right chromosomes. Next, tiny fibers called spindle fibers attach to each chromosome. These fibers are like ropes that will pull the chromosomes apart. The spindle fibers contract, pulling the sister chromatids in opposite directions. The chromosomes split, and what were two arms of each X shaped chromosomes are pulled apart. Once the chromosomes reach the poles of the cell, new nuclear membranes form around each set, forming two new nuclei. Finally, the cytoplasm and the cell membrane divide. The end result, two daughter cells that are genetically identical to the original parent cell. Join us for our next episode, where we will explore stem cells, including what they are and how they can be used in the world of healthcare. Let's start off with exactly what we'll cover in this video. We'll begin by understanding what stem cells are and how they differ from specialized cells. Next, we'll examine the different types of stem cells and where they're found. Then we'll explore the medical applications of stem cells and their potential to treat various diseases. Finally, we'll consider some of the ethical debates surrounding stem cell research. So, what exactly are stem cells? To understand this, we first need to remember what cell differentiation is. Differentiation is the process where a cell becomes specialized for a specific job. For example, some cells differentiate to become muscle cells, while others become nerve cells. Once a cell has differentiated, it generally can't change into a different type of cell. Stem cells, however, are different. They're undifferentiated cells. They have two special abilities that make them unique. First, they can divide to produce more undifferentiated stem cells. Second, they can differentiate into specific specialized cells when given the right signals. This makes them incredibly valuable for medical research and treatments. There are different types of stem cells that can be found in different places. Embryonic stem cells are found in early human embryos, just a few days after fertilization. These cells have the potential to develop into almost any type of cell in the human body. Think about it, every cell in your body, from brain cells to skin cells, originally came from these early embryonic cells. Adults also have stem cells, but they're more limited. Adult stem cells are found in specific areas like bone marrow. Unlike embryonic stem cells, adult stem cells can only differentiate into certain types of cells. For example, bone marrow stem cells can become different types of blood cells, but not nerve cells. Both embryonic and adult stem cells can be grown in laboratories. Scientists can clone them to make genetically identical copies and guide them to differentiate into specific cell types. Stem cells have enormous potential for treating diseases and injuries. Adult stem cells are already being used in medicine today. For example, bone marrow transplants use stem cells to replace faulty blood cells in patients. Embryonic stem cells could also potentially be used to replace damaged or dysfunctional cells in many conditions. They could create new insulin producing cells for people with diabetes, or they might generate new nerve cells for people with spinal cord injuries. To make stem cell treatments even more effective, scientists use a technique called therapeutic cloning. In this process, an embryo is created with the same genetic information as the patient. The stem cells from this embryo would be genetically identical to the patient. This means the patient's immune system wouldn't reject them if they were transplanted. However, there are risks associated with stem cell treatments. For example, stem cells grown in labs might be contaminated with viruses. Stem cell research, particularly embryonic stem cell research, has sparked significant ethical debate. Some people object to embryonic stem cell research on moral grounds. They believe that human embryos deserve protection as potential human lives. They argue that destroying embryos for research violates the sanctity of human life. Others take a different view, focusing on the potential benefits to patients. They point out that embryos used in research are typically extra, unused embryos from fertility clinics. These embryos would likely be discarded if not used for research. They argue that helping existing patients who are suffering should take priority. It's also important to keep in mind that different countries have different regulations regarding stem cell research. In the UK, embryonic stem cell research is permitted but strictly regulated. In some other countries, it's completely banned. Plants also have stem cells located in regions called meristems, which are located at the tips of the roots and shoots of a plant. Meristem cells remain undifferentiated throughout the plant's entire life and can divide and differentiate into any type of plant cell. This ability makes plant stem cells incredibly useful in agriculture and conservation. They can be used to produce identical copies of plants quickly and economically. This technique can help preserve plants that are resistant to certain diseases or plants facing the threat of extinction can be cloned. As you can see, stem cell research represents both remarkable potential and significant challenges. Stick around and watch our next episode, which will focus on diffusion and exchange surfaces. In this lesson, we'll begin by discussing what diffusion is and the factors that affect it, then explore why multicellular organisms need specialized exchange surfaces, examine a range of exchange surfaces in different organisms, and finally, look at how these surfaces are adapted to function efficiently. Let's begin with understanding what diffusion is. Diffusion is the spreading out of particles of any substance in solution, or particles of a gas, resulting in a net movement from an area of higher concentration to an area of lower concentration. This process happens naturally without requiring energy. It's occurring all around us and within our bodies constantly. For example, oxygen and carbon dioxide are exchanged in our lungs by diffusion. Waste products like urea also diffuse from our cells into the blood for excretion by the kidneys. Several factors affect how quickly diffusion happens. The first is the concentration gradient. The bigger the difference in concentration, the faster the rate of diffusion. Temperature also plays a role. Higher temperatures give particles more kinetic energy, causing them to move more rapidly. And finally, the surface area available for diffusion is crucial. The larger the surface area, the faster substances can diffuse. For single celled organisms like amoeba, diffusion works efficiently for all their needs. This is because they have a relatively large surface area compared to their volume. We call this the surface area to volume ratio. However, as organisms become larger and more complex, this ratio decreases. If you imagine a cube getting bigger, its volume increases more rapidly than its surface area. This means that for large multicellular organisms, simple diffusion across the body surface would be far too slow to supply all cells with what they need. This is why larger organisms have evolved specialized exchange surfaces and transport systems. These surfaces allow efficient transfer of materials between the organism and its environment. Let's look at how exchange surfaces in different organisms are adapted for efficient diffusion. All effective exchange surfaces share certain features that help maximize the rate of diffusion. First, they have a large surface area. This increases the amount of material that can be exchanged at any one time. For example, the human lungs contain millions of tiny air sacs called alveoli. These alveoli collectively provide an enormous surface area for gas exchange. Second, exchange surfaces are very thin, providing a short diffusion path. This minimizes the distance that substances need to travel during diffusion. The alveoli walls in our lungs are just one cell thick, allowing gases to pass through quickly. Third, in animals, exchange surfaces have a good blood supply. This maintains a steep concentration gradient by rapidly carrying away substances that have diffused in. It also brings new substances to diffuse out. Our lungs have a dense network of capillaries surrounding each alveolus for this purpose. Fourth, for gas exchange in animals, there's often a mechanism to ventilate the exchange surface. This ensures that fresh supplies of substances are constantly brought to the exchange surface. In humans, this happens through breathing, which brings fresh air into the lungs. Now let's look at specific examples of exchange surfaces in other organisms. In fish, gills serve as the respiratory exchange surface. Water flows over the gill filaments in one direction while blood flows through the capillaries in the opposite direction. This countercurrent flow maximizes the concentration gradient and thus the efficiency of gas exchange. And in plants, the leaves are the main gas exchange surfaces. Leaves have tiny pores called stomata, which can open and close to control gas exchange. Inside the leaf, there are air spaces between cells that increase the surface area available for gas exchange. Going back to the example of animals, they possess a small intestine across which absorption of digested food occurs across the intestinal lining. This lining is folded into finger like projections called villi, which are themselves covered in even smaller microvilli. This arrangement dramatically increases the surface area available for absorption. Understanding diffusion and exchange surfaces helps us appreciate how organisms have evolved to overcome the challenges of size and complexity. These adaptations enable efficient exchange of materials, which is essential for survival. In our next episode, we'll explore another important transport mechanism, osmosis, and how it differs from diffusion, along with the energy requiring process of active transport. Let's start by going over exactly what we'll cover in this video. First, we'll discuss what osmosis is and how it differs from diffusion. Next, we'll examine what active transport is and why it's necessary. And finally, we'll look at specific examples of active transport in plants and animals. Let's begin with understanding osmosis. Osmosis is a specific type of diffusion that involves the movement of water molecules across a partially permeable membrane. Water moves from a region of higher water concentration to a region of lower water concentration. Another way to think about it is that water moves from a dilute solution to a more concentrated solution. This movement continues until the concentration on both sides are equal, or until pressure prevents further movement. The key difference between osmosis and diffusion is that osmosis specifically refers to the movement of water molecules across a membrane. Diffusion, on the other hand, can involve any substance. Water is constantly moving into and out of cells by osmosis. Now let's move on to active transport. Unlike diffusion and osmosis, which are passive processes driven by concentration gradients, active transport requires energy in the form of ATP. Active transport is the movement of substances from a region of lower concentration to a region of higher concentration. This means it works against the concentration gradient. This is why energy is required to work against the natural direction of movement. Why do cells need active transport? There are several reasons. Sometimes cells need to concentrate certain substances inside them, even when these substances are at a lower concentration outside the cell. For example, plant root hair cells need to absorb mineral ions from the soil where these ions are present in very dilute amounts. Additionally, cells might need to remove additional substances completely, maintaining a zero concentration inside the cell. This is the case with certain toxins or waste products. Let's look at specific examples of active transport in organisms. In plants, root hair cells actively transport mineral ions from the soil into the plant. The soil solution is very dilute, containing low concentrations of ions such as nitrates, phosphates, and potassium. For plants to grow, they need to accumulate these ions in much higher concentrations within their cells. This can only be achieved through active transport. In the human digestive system, glucose is absorbed through the small intestine into the bloodstream by active transport. After a meal, glucose concentration in the gut lumen can be lower than in the blood. Against this concentration gradient, energy is used to move glucose into the bloodstream. In summary, passive processes like diffusion and osmosis allow cells to move substances along concentration gradients without using energy. Active transport enables cells to move substances against these gradients. Understanding these transport mechanisms helps us appreciate how cells and organisms maintain their internal environment and interact with their surroundings. These are essential processes for life itself. In our next episode, we will explore more about how cells are organized in organisms in the form of tissues, organs, and organ systems. In this episode, we'll start by understanding the hierarchy of organization from cells to organisms, then explore the different types of tissues found in the body, examine how tissues combine to form organs, and finally see how organs work together in systems to keep organisms functioning. All living organisms are made up of cells, which are the basic units of life. But in complex multicellular organisms like humans, cells don't just exist independently, they're organized into increasingly complex structures. Let's start with the cell. Cells are the basic building blocks of all living organisms. Different cells have different structures that allow them to perform specialized functions. These specialized cells are created through a process called differentiation. This is when cells develop specific features to perform particular tasks. When similar cells work together, they form tissues. A tissue is a group of similar cells that perform a specific function together. In mammals like humans, we have several types of tissues. Muscular tissue is made of cells that can contract, allowing movement. Glandular tissue produces and secretes important substances like hormones and enzymes. And epithelial tissue forms coverings and linings throughout the body. Moving up to the next level of organization, different tissues combine to form organs. An organ is a structure made of multiple tissue types working together to perform specific functions. Take the stomach as an example. It contains muscular tissue that moves food around, glandular tissue that produces digestive enzymes, and epithelial tissue that forms protective linings. At the highest level of organization, organs work together in organ systems. Each organ system performs a particular function necessary for survival. For instance, the digestive system is made up of organs like the stomach, intestines, liver, and pancreas. Each performs a specific role, but together they accomplish the overall function of processing food and absorbing nutrients. The digestive system includes glands like the pancreas and salivary glands that produce digestive enzymes, while the main function of the stomach and small intestine is to break down and digest food, and the liver produces bile to help with fat digestion. And finally, the large intestine absorbs water from undigested food, leaving behind the final waste products. All our organ systems, such as digestive, respiratory, circulatory, and others, work together to form a complete functioning organism. This organization from cells to organism allows for specialization and efficiency. Each level performs specific tasks while contributing to the overall functioning of the body. In this lesson, we'll begin by looking at what enzymes are and why living organisms need them, then examine their structure and how they catalyze specific reactions, learn about the different digestive enzymes and where they're produced, And finally, follow food through the digestive system to see how enzymes break it down into usable molecules. Chemical reactions are essential for life. They build the molecules your body needs, break down food for energy, and allow your cells to communicate. But these reactions would be far too slow without help. This is where enzymes come in. Enzymes are biological catalysts produced by living organisms. A catalyst is a substance that speeds up a chemical reaction without being changed or used up itself. Living things have thousands of different chemical reactions happening simultaneously inside them. All these reactions need to be carefully controlled. One way to speed up reactions is by raising the temperature, but this approach has major limitations for living organisms. High temperatures would accelerate all reactions indiscriminately, including harmful ones. So instead, living organisms produce enzymes, specialized proteins that can speed up specific reactions at body temperature. This allows for precise control of which reactions happen and when. Enzymes are large protein molecules made up of chains of amino acids. These chains fold into specific three-dimensional shapes that are crucial for enzyme function. The most important feature of an enzyme is its active site, a special region where the chemical reaction takes place. The active site has a unique shape that allows it to bind to a specific substrate molecule, the substance the enzyme acts upon. Enzymes are highly specific. Each enzyme typically catalyzes just one type of reaction. This specificity comes from the precise shape of the active site, which only allows certain molecules to fit, similar to how a key fits into a lock. This lock and key model helps explain enzyme specificity, though the actual mechanism is slightly more sophisticated. When a substrate enters the active site, the enzyme adjusts its shape slightly to create an even better fit, a process called induced fit. Temperature and pH greatly affect how well enzymes work. Each enzyme has an optimal temperature and pH range where it functions most effectively. As temperature increases, enzyme activity initially increases because molecules move faster, creating more opportunities for enzymes and substrates to collide. However, if the temperature gets too high, the heat damages the bonds that maintain the enzyme's three-dimensional structure. This changes the shape of the active site, and the enzyme can no longer function properly. We say the enzyme has been denatured. Similarly, pH affects enzyme structure and function. Extreme pH levels can change the shape of an enzyme's active site by altering the chemical bonds that maintain its structure. Each enzyme has an optimal pH where it works best. For example, digestive enzymes in the stomach function best in highly acidic conditions, while enzymes in the small intestine prefer alkaline environments. Now let's see how enzymes are put to work in the digestive system, breaking down the food you eat into nutrients your body can use. The food you eat contains nutrients essential for life, but most of these nutrients are locked in large, complex molecules that your body can't directly use. These molecules must be broken down into smaller components that can be absorbed into your bloodstream with the help of digestive enzymes. There are three main types of digestive enzymes, each targeting a specific type of nutrient. Carbohydrates break down carbohydrates like starch into simple sugars such as glucose and maltose. Amylase is the main carbohydrate in your digestive system. It's produced in three different places: the salivary glands in your mouth, the pancreas, and the small intestine. Proteases break down proteins into amino acids. The stomach produces a protease called pepsin. Additional proteases are made by the pancreas and the small intestine. Lipases break down fats, also called lipids, into fatty acids and glycerol. These enzymes are primarily produced by the pancreas and the small intestine. Once these large molecules are broken down into their smaller components, sugars, amino acids, fatty acids, and glycerol, they can easily pass through the walls of your digestive system into the bloodstream. From there, they're transported to cells throughout your body where they can be used for energy or to build new tissues. While not an enzyme itself, bile plays a crucial role in the digestive process. Bile is produced by the liver and stored in the gallbladder until needed. It serves two important functions. First, food leaving the stomach is highly acidic due to stomach acid, but enzymes in the small intestine work best in alkaline conditions. Bile helps neutralize this acid, creating the optimal environment for these enzymes to function. Second, bile helps emulsify fats, breaking large fat droplets into tiny ones, similar to how dish soap breaks up grease. This significantly increases the surface area of fat exposed to lipase enzymes, dramatically accelerating fat digestion. The digestive system demonstrates the incredible efficiency of enzyme catalyzed reactions. Without these specialized proteins, we couldn't extract the nutrients we need from food. In our next episode, we'll explore another vital system, the respiratory system, and how it provides the oxygen needed for cellular respiration. In this episode, we will focus on the lungs and the heart, two vital organs that ensure oxygen reaches every cell in your body. First, we'll examine the structure of the lungs and how they enable gas exchange. Next, we'll explore the path of air through the respiratory system. Then we'll take a look at the heart and its role in the circulatory system. And finally, we'll examine how the heart pumps blood through the double circulation system. Your cells need a constant supply of oxygen to carry out respiration, the process that releases energy from food molecules. At the same time, they need to eliminate carbon dioxide, a waste product of respiration. The respiratory system is specialized to take in oxygen and remove carbon dioxide, while the circulatory system transports these gases throughout the body. Let's start by understanding the structure of the respiratory system. This system uses lungs, which are located in the thorax, which is in the upper part of your body. They're protected by the ribcage and separated from the lower body by the diaphragm. The lungs themselves are soft, spongy organs that appear pink and are surrounded by a protective membrane called the pleural membrane. When you breathe in, air enters your body through your nose or mouth and travels down the trachea. The trachea then splits into two tubes called bronchi, one leading to each lung. Inside the lungs, the bronchi continue to branch into progressively smaller tubes called bronchioles, similar to how a tree branches into smaller and smaller limbs. These bronchioles eventually end at tiny air sacs called alveoli. Alveoli are where the actual gas exchange takes place. The lungs contain millions of these microscopic air sacs, each surrounded by a network of tiny blood vessels called capillaries. The walls of both the alveoli and capillaries are extremely thin, just one cell thick. This allows gases to diffuse quickly between the air in the alveoli and the blood in the capillaries. When blood arrives at the lungs, it's returning from the body's tissues where it has picked up carbon dioxide and released oxygen. This blood is low in oxygen and high in carbon dioxide. Inside the alveoli, the oxygen concentration is high because of the air you've just breathed in, while carbon dioxide concentration is low. Following the principle of diffusion, gases move from areas of high concentration to areas of low concentration. So oxygen diffuses from the air in the alveoli into the bloodstream, while carbon dioxide diffuses from the blood into the alveoli to be exhaled. The oxygen that enters the bloodstream binds to the hemoglobin molecules inside red blood cells. Hemoglobin is a protein which gives blood its red color and is specialized for oxygen transport. But getting oxygen into the blood is only half the story. The circulatory system needs to transport this oxygen to all the cells in the body, and at the center of the system is the heart. The circulatory system is the body's transport network. It delivers essential oxygen and nutrients to every cell in your body and removes waste products like carbon dioxide. Think of it as a delivery system and a waste collection system. This system consists of three main components: the heart, which acts as a pump, the blood vessels, which form the network of tubes through which the blood travels, and the blood itself, which carries the materials throughout the body. Humans have what's called a double circulatory system. This means we have two separate circuits of blood flow, both connected to the heart. The first circuit is called pulmonary circulation. In this circuit, deoxygenated blood is pumped from the right side of the heart to the lungs. In the lungs, this blood picks up oxygen and releases carbon dioxide through the alveoli, as we just discussed. The newly oxygenated blood then returns to the left side of the heart. The second circuit is called systemic circulation. The oxygenated blood is pumped from the left side of the heart to all the other organs and tissues of the body. The blood delivers oxygen and nutrients to these tissues and picks up carbon dioxide and other waste products. The deoxygenated blood then returns to the right side of the heart, completing the cycle. At the center of the system is the heart, a remarkable organ about the size of your fist. The heart is essentially a muscular pump that keeps blood moving through the blood vessels. The walls of the heart contract rhythmically on their own without conscious control. This allows your heart to beat continuously throughout your life. The heart has four chambers: two upper chambers called atria, and two lower chambers called ventricles. The right atrium and right ventricle form the right side of the heart, which pumps deoxygenated blood to the lungs. The left atrium and left ventricle form the left side, which pumps oxygenated blood to the rest of the body. Between these chambers are special one-way valves that ensure blood flows in only one direction, preventing any backflow. Here's how blood flows through the heart. First, deoxygenated blood enters the right atrium through a vein called the vena cava. The right atrium contracts, pushing this blood into the right ventricle. When the right ventricle contracts, it forces blood into the pulmonary artery, which carries it to the lungs. After picking up oxygen in the lungs, the blood returns to the heart through the pulmonary veins, entering the left atrium. The left atrium contracts, pushing blood into the left ventricle. Finally, the powerful left ventricle contracts, forcing blood into the aorta, the largest artery in the body, which distributes it to all the other organs and tissues. Interestingly, the heart itself needs its own blood supply. Coronary arteries branch off the aorta and wrap around the heart, delivering oxygen and nutrients at the heart muscles, also known as cardiac muscles, need to function. The rhythmic beating of the heart is controlled by a natural pacemaker, a specialized group of cells located in the wall of the right atrium. These cells generate electrical impulses that spread through the heart muscle, causing it to contract in a coordinated manner. If this natural pacemaker malfunctions, an artificial pacemaker may be implanted. This small electronic device generates electrical impulses to regulate heartbeat. The respiratory and circulatory systems work together to ensure that oxygen reaches every cell in your body and that carbon dioxide is efficiently removed. This partnership is essential for life. Without it, cells would quickly run out of oxygen and energy, and toxic waste products would build up. In our next episode, we'll continue exploring the circulatory system by looking at the different types of blood vessels and the components of blood. In this lesson, we'll begin by exploring the three types of blood vessels and their specialized structures, then examine how these vessels work together to form a complete circuit, look at the components of blood and their specific functions, and finally see how blood transports substances and defends the body against disease. Blood vessels form an extensive network that reaches every cell in your body. This network contains three types of vessels, each with a specialized structure suited to its function. The three types of blood vessels are arteries, capillaries, and veins. Each has unique characteristics that allow it to perform its specific role in the circulatory system. Arteries carry blood away from the heart under high pressure. The heart's powerful contractions create this pressure, forcing blood to move quickly through the arterial system. Because arteries must withstand this high pressure, they have thick, strong, and elastic walls. These walls contain multiple layers, including a thick layer of smooth muscle and elastic fibers. The central channel through which blood flows, called the lumen, is relatively small compared to the thickness of the arterial wall. This helps maintain the high pressure needed to push blood throughout the body. As arteries move further from the heart, they branch into progressively smaller vessels. Capillaries are the smallest blood vessels in the body, so small they can only be seen under a microscope. Despite their tiny size, capillaries are perhaps the most important part of the circulatory system. They're where the actual exchange of substances between blood and tissues occurs. Several features make capillaries perfectly adapted for this exchange function. First, they're extremely numerous and form extensive networks that reach every cell in the body. Second, capillary walls are incredibly thin, usually just one cell thick. This minimizes the distance substances must diffuse to travel between the blood and surrounding tissues. Third, these thin walls are permeable, allowing small molecules like oxygen, carbon dioxide, nutrients, and waste products to pass through easily. After passing through capillaries, blood flows into veins, which return it to the heart. By the time blood reaches the veins, it's under much lower pressure than in arteries. This presents a challenge: how to keep blood flowing back to the heart against gravity, especially from the lower parts of the body. Veins have several adaptations to overcome this challenge. First, they have a larger lumen or internal space than arteries of comparable size. This larger channel offers less resistance to blood flow, helping blood move despite the lower pressure. Second, many veins, especially those in the limbs, contain valves. These one-way valves prevent blood from flowing backward due to gravity. Third, vein walls are thinner than artery walls since they don't need to withstand high pressure. However, they still contain some muscle and elastic tissue to maintain their shape and assist with blood flow. Flowing through this complex network of vessels is blood, a specialized tissue that transports vital substances throughout the body. Blood is classified as a tissue even though it's fluid, and it makes up about 7 to 8% of your body weight. Blood consists of four main components: red blood cells, white blood cells, platelets, and plasma. Each component has specific functions essential for survival. Let's start with red blood cells. These are the most numerous cells in your blood and give blood its red color. The primary function of red blood cells is to transport oxygen from your lungs to all the cells in your body. They also help carry some carbon dioxide back to the lungs. Red blood cells have several adaptations that make them perfect for this job. They have a distinctive biconcave disc shape, like a donut with a thin center instead of a hole. This unique shape provides a large surface area relative to volume, allowing for efficient gas exchange. Unlike most cells in your body, red blood cells have no nucleus. This allows more space for hemoglobin, the iron containing protein that binds to oxygen. In the lungs, oxygen binds to this protein, forming oxyhemoglobin. When the blood reaches body tissues where oxygen concentration is low, the oxygen separates from hemoglobin and diffuses into the cells where it's needed for cellular respiration. The next component is white blood cells. These are your body's defense force against disease and infection. While far less numerous than red blood cells, white blood cells are critical for your immune system. There are several types, each with specific roles in protecting your body. Some white blood cells, called phagocytes, can change their shape to surround and engulf harmful microorganisms in a process called phagocytosis, essentially eating the invaders. Others, called lymphocytes, produce antibodies, specialized proteins that identify and neutralize foreign substances like bacteria and viruses. White blood cells also produce antitoxins that neutralize toxins released by certain bacteria. The third component, platelets, are not complete cells but small cell fragments that don't have a nucleus. Despite their simple structure, platelets serve a vital function. When you cut yourself, platelets help form blood clots to stop bleeding. Without this clotting mechanism, even a small cut could lead to dangerous blood loss. Platelets gather at the site of injury and stick together, forming a plug that helps seal the wound. People with low platelet counts may experience excessive bleeding or bruising, highlighting the importance of these tiny cell fragments. The final component is plasma, the liquid portion of blood. Plasma makes up about 55% of your blood volume and is mostly water, with a pale, straw like color. Plasma serves as the transport medium for all the other components of blood. It carries red and white blood cells and platelets throughout your body. It also transports a variety of dissolved substances, including glucose, amino acids, carbon dioxide, and hormones. Together, these blood vessels and blood components form an extraordinary transport system. Let's start by going over exactly what we'll cover in this video. We'll begin by understanding what coronary heart disease is and how it develops. Next, we'll look at how stents are used to keep coronary arteries open and restore blood flow. Then we'll explore the role of statins in reducing blood cholesterol levels. We'll also discuss mechanical devices and heart transplants for treating severe heart failure. And finally, we'll examine how damaged heart valves can be replaced using biological or mechanical alternatives, along with blood substitutes in cases of major blood loss. Coronary heart disease is a major health issue that affects many people. It occurs when the coronary arteries, which supply blood to the heart muscle, become narrowed. This narrowing happens because fatty materials build up inside these arteries, forming layers that reduce the flow of blood. When the heart muscle doesn't get enough oxygen from the blood, it can't function properly. As these fatty deposits increase over time, the space inside the arteries gets smaller and smaller. This means less and less blood can flow through to deliver oxygen to the heart muscle. If blood flow becomes severely restricted, a person may experience chest pain. In a worst case scenario, if an artery becomes completely blocked, part of the heart muscle can die, resulting in a heart attack. One effective treatment for narrowed coronary arteries is the use of stents. Stents are small, tube-shaped devices that are placed inside affected arteries. These clever devices physically hold the blood vessel open, allowing blood to flow through more easily. This ensures the heart muscle receives the oxygen it needs to function properly. Stents offer several advantages for patients. They're relatively non-invasive compared to major heart surgery, and recovery times are typically shorter. Once in place, they provide long-term benefits by keeping the artery open. However, stent procedures do come with some risks. There's a small chance of complications during the procedure itself. After placement, there's a risk of blood clots forming around the stent, causing thrombosis. Another important treatment for coronary heart disease involves medications called statins. These drugs help to reduce the levels of cholesterol in the blood. Cholesterol is a fatty substance that's actually essential for our bodies. We need it to build cell membranes and make certain hormones. However, when we have too much of the wrong type of cholesterol, it can contribute to those fatty deposits in our arteries. Statins work by reducing how much cholesterol our body produces, particularly the harmful type known as LDL cholesterol. By lowering these levels, statins slow down the buildup of fatty deposits in the arteries. For many patients, taking statins regularly can significantly reduce their risk of heart attacks and stroke. These medications may also help to stabilize existing deposits. preventing them from suddenly breaking off and blocking an artery completely. Like all medications, statins can have side effects. Some people experience muscle aches, headaches, or digestive issues. In rare cases, they can cause more serious problems with the liver or muscles. Another con of using statins is that they can take weeks or months to have any noticeable effect, and a patient will need to continue taking them to keep receiving the health benefits. If they stop, the condition will likely continue to worsen. In cases where a heart attack has occurred, more drastic interventions may be necessary. Sometimes the heart becomes so weakened that it can no longer pump blood effectively. For patients with severe heart failure, a heart transplant might be the best option. This involves replacing the damaged heart with a healthy donor heart. While this can be life-saving, finding suitable donor hearts is challenging, and patients may wait months or years. During this waiting period, or for patients who aren't suitable transplant candidates, mechanical heart devices can provide a solution. These devices help the weakened heart pump blood more effectively. Some mechanical devices are designed to support the heart temporarily until a donor heart becomes available. Others may be used as a long-term solution when transplantation isn't possible. The advantage of mechanical devices is they're immediately available and don't trigger immune rejection. However, they require major surgery to implant, and patients often need to take blood-thinning medications to prevent clots from forming. Heart valves are another part of the heart that can be damaged. These valves control the direction of blood flow through the heart, ensuring it moves forward efficiently. When heart valves become damaged or diseased by old age or a heart attack, they may not open fully or they may be unable to fully close. Either way, the heart has to work harder to pump the same amount of blood. Faulty heart valves can be replaced in surgery. Surgeons might use biological valves, which come from human donors or animals. Alternatively, they may use mechanical valves made from durable synthetic materials. Biological valves work very naturally, but typically need replacing after a certain number of years. Mechanical valves last longer, but require patients to take blood-thinning medication for life to prevent clots from forming on the valve. In situations involving major surgery or injury, patients may experience significant blood loss. While blood transfusions are the standard treatment, there are times when donated blood isn't available or can't be used. This is where blood substitutes become important. Blood substitutes are artificial substances designed to perform some of the functions of blood, particularly carrying oxygen to tissues. Unlike donated blood, these substitutes don't require blood type matching and have a longer shelf life. Currently available blood substitutes include saline-based solutions. These can be used in emergency situations to stabilize patients until proper blood transfusions become available or until their body produces more blood cells to replace the lost blood. Our understanding of heart disease and its treatments has advanced tremendously over recent decades. What was once a death sentence can now often be managed effectively, allowing patients to lead full and active lives. Today, we're learning about what health really means and how it relates to disease, then looking at the difference between communicable and non communicable diseases, exploring how different types of diseases can interact with each other, and finally, seeing how our lifestyle choices can affect our health. So, what is health? Health isn't just about not being ill, it's about feeling good physically and mentally. The World Health Organization defines health as a state of complete physical, mental, and social well being. Diseases are a major cause of ill health. A disease is any condition that stops the body from working normally. Diseases can be split into two main types: communicable and non communicable. Communicable diseases can spread from one person to another. They're caused by pathogens, things like bacteria, viruses, fungi, and protists. Examples include flu, measles, and food poisoning. Non communicable diseases can't spread between people. They often develop slowly over time. Examples include asthma, diabetes, and heart disease. These are often linked to your genes, your lifestyle, or your environment. Now, here's something really important to understand. Different types of diseases can interact with each other in ways that make your health worse. Let's look at four key ways this can happen. First, if your immune system isn't working properly, a non communicable issue, then you're more likely to catch infectious or communicable diseases. Your immune system is like your body's defense team. If it's weakened, pathogens can invade more easily. For example, someone with HIV has a weakened immune system, making them more vulnerable to infections. Second, some viruses can actually cause cancer. For example, certain types of human papillomaviruses can lead to cervical cancer. This shows how a communicable disease can sometimes lead to a non communicable disease. Third, infections can trigger allergic reactions. For example, if you have asthma and catch a cold, your asthma symptoms might get worse. The infection triggers your immune system, which can cause inflammation in your airways. Fourth, physical illness can affect your mental health. If you're dealing with a serious or long term physical illness, you might develop depression or anxiety. This shows how physical and mental health are connected. It's also worth remembering that health is affected by many other factors besides disease. What you eat, how much stress you have, how active you are, and whether you can see a doctor when you need to, all these things matter too. For example, eating a balanced diet gives your body what it needs to stay healthy. Too much stress can lead to problems like high blood pressure, and seeing a doctor regularly means health problems can be spotted early. Understanding how different aspects of health are connected helps doctors take a more rounded approach to keeping people healthy and treating disease. Today, we're learning about what risk factors are and why they matter, then looking at different types of risk factors that can make us more likely to get certain diseases, seeing how these factors affect people differently depending on where they live, and finally, exploring why this matters for individuals, families, and society. So, what exactly is a risk factor? Simply put, it's something that makes you more likely to develop a particular disease. But here's an important point. Having risk factors doesn't mean you'll definitely get a disease. It just increases your chances. Think of it like this. If you don't revise for an exam, you have a higher risk of failing. But some people might still pass without revising. Risk factors come in different types. Some are related to your lifestyle, like what you eat, how much you exercise, or whether you smoke. Others are environmental, like pollution in the air or chemicals you might be exposed to. Some risk factors you can't control, like your age or family history, but many others you can change. What makes non communicable diseases tricky is that they're usually caused by several risk factors working together, not just one thing. Risk factors affect people differently around the world. Let's look at the global, national, and local levels. Globally, in wealthier countries, people tend to have more non communicable diseases like heart disease and diabetes. This is often because people can afford fast food, have more sedentary jobs, and drive rather than walk. In less developed countries, people might have different health problems, though non communicable diseases are increasing there too. At the national level, even within the UK, there are big differences between regions. Some areas have higher rates of smoking or obesity than others, often linked to factors like income levels and education. Government policies on things like cigarette prices and food labeling can make a big difference nationally. Locally, even neighborhoods in the same city can have very different health patterns. Some local areas might have lots of fast food shops, but few parks or sports facilities. Other neighborhoods might have more air pollution if they're near busy roads or factories. These local factors combine to create hotspots, where certain diseases are much more common. Some risk factors directly cause diseases. Smoking is the clearest example. The chemicals in cigarettes directly damage your lungs and blood vessels. When you smoke, tar coats your lungs and toxic chemicals enter your bloodstream. damaging cells throughout your body. Carrying too much body fat can lead to diabetes because it affects how your body responds to insulin. Your cells become less sensitive to insulin, making it harder for your body to control blood sugar levels. Drinking too much alcohol directly harms your liver, which has to work overtime to process the alcohol in your blood. Over time, this can cause liver cells to die and be replaced by scar tissue. During pregnancy, smoking or drinking alcohol can directly harm the developing baby. Scientists find risk factors by looking for patterns in data, but it's important to understand the difference between correlation and causation. Just because two things happen together doesn't mean one causes the other. For example, not exercising and eating lots of fatty foods are linked to heart disease, but these behaviors don't directly damage your heart. They cause high blood pressure and cholesterol, which then damage your arteries. Non communicable diseases have huge impacts on people's lives. Millions die each year from these diseases, and many more have a poorer quality of life. These diseases also cost a lot of money, both for the healthcare system and for families affected by them. Healthcare systems spend billions on treating conditions that could potentially be prevented. Families might need to adapt their homes or reduce working hours to care for someone with a chronic illness. If someone can't work because of illness, their family might struggle financially. When lots of people in a country are ill, it affects the whole economy. Fewer people working means less money being made. Understanding risk factors is super useful because it helps us make better choices for our health. Making small, sustainable changes to our lifestyle can have a big impact on our health, both now and in the future. In this video, we're learning about what cancer actually is and how it develops in the body, then exploring the difference between benign and malignant tumors, examining the various risk factors that can increase someone's chance of developing cancer, and finally seeing how lifestyle choices and genetics can play a role in cancer development. So, what exactly is cancer? Simply put, cancer is caused by uncontrolled cell growth and division. Normally, our cells grow and divide in a controlled way, but sometimes changes occur in the cells that disrupt this control system. When this happens, cells begin to grow and divide too much, forming a lump of cells called a tumor. Not all tumors are cancerous, though. They can be either benign or malignant. Benign tumors stay in one place, usually within a membrane, rather than invading other tissues. They don't normally spread to other parts of the body and aren't usually dangerous. Malignant tumors, however, grow and spread to neighboring healthy tissues. Cancer cells can break off and spread to other parts of the body through the bloodstream. These malignant cells then invade healthy tissues elsewhere and form secondary tumors. This ability to spread makes malignant tumors dangerous and potentially fatal. Various risk factors can increase the chance of developing certain types of cancer. It's important to understand that having risk factors doesn't mean you will definitely get cancer. Scientists have identified lots of lifestyle risk factors linked to different types of cancer. Smoking is perhaps the most well known. It's strongly linked to lung cancer. But research shows smoking can cause other types of cancer too, including mouth, throat, stomach, and cervical cancer. Obesity has been linked to many different cancers, including those of the bowel, liver, and kidney. In fact, obesity is the second biggest preventable cause of cancer after smoking. UV exposure from the sun or sunbeds increases your risk of developing skin cancer. People who live in sunny climates or spend a lot of time outside without protection face higher risks. Using sunscreen regularly and covering up can help reduce this risk. Viral infections with certain viruses have been shown to increase the chances of developing specific cancers. For example, hepatitis B and hepatitis C viruses can increase the risk of liver cancer. Risk factors can also be associated with genetics. Sometimes you can inherit faulty genes that make you more susceptible to cancer. For example, mutations in the BRCA genes have been linked to an increased likelihood of developing breast and ovarian cancer. Regular screening can also help detect cancer early when treatment is most likely to be effective. Join us for our next episode where we will learn about tissue organization in plants. Today we're learning about plant tissues, how they form organs like leaves, roots, and stems, exploring leaf structure in detail, and finally discovering how these structures enable transport and photosynthesis. Similar to how animal bodies have various specialized cells working in coordination, plants contain different types of cells that collaborate to form functional tissues. Plant tissues include several types: epidermal tissue, palisade mesophyll, spongy mesophyll, tissue for transportation such as xylem and phloem, and finally meristem tissue. The outermost layer of plants is composed of epidermal tissue, which serves as a protective barrier for the tissues beneath. This defensive layer features a waxy coating called the cuticle, which helps minimize water loss and offers protection against potential pathogens. Just beneath the upper surface of leaves lies the palisade mesophyll layer. These densely arranged cells contain numerous chloroplasts, positioning this region as the key location where most photosynthesis takes place. Deeper within the leaf structure is the spongy mesophyll layer. This tissue consists of irregularly shaped cells separated by air pockets. These air spaces facilitate the movement of carbon dioxide and oxygen to and from the cells, supporting the leaf's respiratory functions. Throughout plant structures run two vital transportation tissues, xylem and phloem. Xylem vessels function like pipelines, moving water and dissolved nutrients from the roots upward to the stems and foliage. These cellular structures create hollow channels reinforced with lignin, enabling them to efficiently transport water through the plant's vascular system. Phloem tissue manages the distribution of sugars produced during photosynthesis, transporting them from the leaves to other parts of the plant. This movement of sugar molecules through the phloem network is technically known as translocation. At growth points of shoots and roots, we find meristem tissue. These undifferentiated cells possess the ability to divide and generate new cellular material, facilitating plant growth and development. A remarkable characteristic of meristem tissue is its capacity to develop into any type of plant cell throughout the plant's lifetime. The leaf functions as a specialized plant organ optimized for photosynthesis. Its broad surface captures maximum sunlight, while its thin profile allows efficient gas exchange. The lower epidermis contains tiny openings called stomata, which enable gases to move into and out of the leaf interior. Each stomatal opening is flanked by a pair of specialized guard cells that can modify their shape to regulate the size of the pore. This mechanism controls gas exchange rates and helps limit excessive water loss through transpiration. The evolution of plants has resulted in remarkably efficient systems, from individual specialized cells to complex tissues to functional organs, enabling plants to thrive in diverse environmental conditions. Today we're learning about xylem and phloem transport systems, how transpiration drives water movement through plants, how factors like light, temperature, airflow, and humidity affect transpiration rates, and finally, how guard cells control stomata to regulate gas exchange and water loss. Despite what you might imagine, plants don't have tiny vehicles moving substances around inside them, but they do have fascinating transport systems. Let's start with phloem tubes, which transport food throughout the plant. Phloem consists of arrangements of living cells that have specialized end walls with small openings or pores. These openings allow the contents to move from one cell to another. Phloem's primary function is to move food substances, especially the sugars produced in the leaves, to other parts of the plant. This nutrient transport provides energy for growing areas or gets stored for later use. Interestingly, this movement can occur both upward and downward in the plant, depending on where nutrients are needed. The technical term for this movement of food through phloem is translocation. Now let's examine xylem tubes, which are responsible for water transport. Unlike phloem, xylem is composed of non-living cells connected together without separating walls, creating what's essentially a continuous pipeline. These xylem vessels are reinforced with a substance called lignin that provides strength and rigidity to maintain their structure. Xylem transports water containing dissolved minerals from the roots upward to the stems and leaves. This continuous flow of water from roots to leaves is known as the transpiration stream. So, what exactly is transpiration? In simple terms, it's the process by which plants lose water to the atmosphere. But the process is more complex than it first appears. Transpiration occurs through evaporation and diffusion of water from the plant's surfaces. The majority of water loss happens through the leaves via small openings called stomata. When water evaporates from the leaf cells, it creates a mild water deficit, which pulls more water up from other parts of the plant through the xylem vessels. This in turn draws more water up from the roots, creating a continuous flow of water moving through the entire plant. An interesting point to note is that transpiration isn't actually the plant's goal. It's a consequence of the leaf structure needed for photosynthesis. Plants must have stomata to allow carbon dioxide to enter for photosynthesis. However, the concentration gradient means that water inside the leaf will inevitably escape through these same openings. This water loss is a necessary compromise for the plant to carry out photosynthesis. Now let's investigate the factors that influence how quickly plants lose water. Four main variables affect transpiration rates. Light conditions significantly impact transpiration. More intense light leads to increased water loss. This occurs because stomata tend to close in darkness. Since photosynthesis cannot occur without light, plants don't need to keep their stomata open for carbon dioxide intake in the dark. With closed stomata, minimal water escapes from the plant. Environmental temperature also plays a crucial role. Warmer conditions accelerate transpiration. This happens because heat gives water molecules additional energy to evaporate and move out through the stomata. Air movement around the plant is another important factor. Stronger air currents increase water loss. When air is still around a leaf, evaporated water accumulates near the leaf surface. This creates similar water vapor concentrations inside and outside the leaf, slowing further diffusion. However, when air currents remove this moisture layer, the concentration gradient is maintained, allowing continuous, rapid diffusion from high to low concentration. The fourth factor is atmospheric moisture. Drier surrounding air accelerates transpiration. Scientists can measure transpiration rates by monitoring water uptake by plants. This approach works because the amount of water absorbed by the plant correlates directly with water lost through leaf transpiration. To conduct such measurements, researchers use equipment featuring a plant with roots in water, a narrow tube, and a method to track air bubble movement in the tube. By noting the initial position of the bubble and the distance it travels over time, one can determine the rate of water absorption. For accurate results, it's essential to maintain consistent experimental conditions, particularly regarding temperature and humidity levels. Let's now examine guard cells, which regulate stomatal openings. These specialized cells have a distinctive curved shape that enables them to control the size of the stomatal pore. When guard cells absorb water, they become swollen and firm. This change in shape creates an opening between them for gas exchange during photosynthesis. During water shortages, guard cells release water and become less rigid, causing the stomata to close. This mechanism helps prevent excessive moisture loss from the plant. The ability of guard cells to change shape comes from their unique structure. They feature thinner walls on their outer sides and thicker walls on their inner surfaces, which influences how they deform when their water content changes. In conclusion, plants have evolved remarkable transport mechanisms for moving water, minerals, and nutrients to where they're needed. Today, we're learning about various disease causing organisms and their distinctive characteristics, how these organisms make us ill through different mechanisms, the various transmission methods that allow infections to pass from one host to another, and finally, how these infections affect both the plant and animal kingdoms. Disease causing microorganisms, known as pathogens, invade the body and trigger illness. These microscopic invaders come in four main categories: bacteria, viruses, protists, and fungi. Each operates differently when causing sickness. Bacteria are microscopic single celled organisms. Despite their tiny dimensions, they multiply quickly once inside the human body. What makes bacterial infections problematic is that many bacteria release harmful chemicals as they grow. These substances interfere with normal cell function and harm tissues, resulting in the symptoms we experience during infection. Viruses are considerably smaller than bacteria, approximately 1/100th of a bacterial cell's size. Unlike bacteria, viruses lack cellular structure, consisting merely of genetic material encased in protein. Unable to multiply independently, viruses hijack the host cell's internal mechanisms. They force our cells to create viral components before assembling numerous new virus particles. Eventually, the infected cell typically ruptures, releasing newly formed viruses that infect neighboring cells. This destruction explains many symptoms associated with viral infections. Moving to protists, these are more complex, single celled organisms containing a nucleus. Most disease causing protists function as parasites, residing on or within hosts while causing cellular disruption. Many protist infections reach humans through carrier organisms called vectors. These carriers transport the pathogen without becoming ill themselves. Our final pathogen category, fungi, ranges from single celled organisms to complex structures formed of thread like filaments called hyphae. These filaments can extend into human skin or plant surfaces, establishing infection. Many fungi distribute through reproductive particles called spores, enabling the infection to spread between hosts. Regarding transmission, pathogens spread through three primary routes. The first involves water, where organisms contaminate drinking sources or recreational waters. For instance, certain bacterial infections occur when people consume water containing waste from infected individuals. Airborne transmission forms our second route. Some pathogens travel through air currents, particularly in moisture droplets released during respiratory actions like coughing. Respiratory infections commonly spread this way. Our third transmission method involves direct contact. Numerous pathogens transfer through touching contaminated objects or infected individuals. Many skin infections propagate through shared surfaces in communal areas. It's worth noting that pathogens don't exclusively target humans. Plants and animals also suffer from various infections. These can significantly impact growth, reproduction, and survival across all living kingdoms. Today, we're learning about three viral diseases: measles, HIV, and tobacco mosaic virus, then looking at two bacterial infections, salmonella and gonorrhea, studying a fungal disease called rose black spot, and finally investigating malaria, a disease caused by a protist. Let's start with viral diseases. Measles is a viral infection that causes fever and a distinctive red skin rash. It spreads through droplets in the air when an infected person sneezes or coughs. These tiny moisture droplets can be inhaled by others nearby. People with measles typically develop a high temperature and a red rash that starts on the face and spreads across the body. Measles can be serious or even fatal if complications develop. It can lead to pneumonia, which is inflammation of the lungs, or encephalitis, which is inflammation of the brain. Because of these serious risks, most children receive vaccination against measles when they're young, which has helped reduce cases worldwide. The second viral disease we'll examine is HIV, which stands for Human Immunodeficiency Virus. HIV spreads through sexual contact or by exchanging bodily fluids, such as blood. This often happens when people share needles during drug use. When first infected with HIV, a person may experience flu like symptoms for a short period. They might then have no symptoms at all for several years. During this time, however, the virus continues to attack the immune system. It specifically targets immune cells, gradually weakening the body's natural defenses. Without treatment with antiretroviral drugs, the immune system becomes severely damaged, leading to AIDS, which stands for Acquired Immune Deficiency Syndrome. At this stage, the body cannot fight off other infections or cancers effectively. People with AIDS often develop infections that wouldn't affect someone with a healthy immune system. Viruses don't just affect humans, they can infect plants too. Tobacco mosaic virus, which is also known as TMV, affects many plant species, including tomatoes. It causes a distinctive mottled pattern on the leaves where some areas appear discolored. This pattern gives the virus its name. The discoloration means the plant can't photosynthesize properly, which affects growth. Infected plants often produce fewer fruits and may be stunted. TMV spreads when plants come into contact with each other or through gardening tools. Unlike animal viruses, it doesn't need vectors like insects to spread. Now let's move to bacterial diseases. Salmonella is a type of bacteria that causes food poisoning. People infected with salmonella experience fever, stomach cramps, vomiting, and diarrhea. These symptoms usually appear within a day or two of infection. These symptoms occur because the bacteria produce toxins that irritate the digestive system. You can get salmonella by eating food contaminated with the bacteria. This includes undercooked meat, eggs from infected birds, or food that's been cross contaminated during preparation. In the UK, most poultry are vaccinated against salmonella to help control its spread. This public health measure has reduced human infections significantly. The second bacterial disease we'll look at is gonorrhea, a sexually transmitted infection. Unlike salmonella, gonorrhea spreads through sexual contact, especially unprotected sex. A person with gonorrhea will often feel pain when they urinate. Another common symptom is a thick, yellow, or green discharge from the affected areas. Gonorrhea used to be easily treated with penicillin. However, many strains of the bacteria have become resistant to this antibiotic. This antibiotic resistance is a growing concern in treating bacterial infections. It happens when bacteria evolve in response to the drugs used against them. To prevent gonorrhea spreading, people can use barrier methods of contraception like condoms, and those infected can be treated with appropriate antibiotics. Now let's take a look at fungal diseases. Rose black spot is a common fungal disease affecting rose plants. This fungus causes purple or black spots to develop on the leaves of rose plants. The spots have irregular edges and can grow quite large. As the disease progresses, affected leaves often turn yellow and fall off prematurely. This early leaf loss weakens the plant. When the plant loses its leaves, it can't photosynthesize as effectively. This means less energy for growth and flowering. The fungus spreads through the environment by water or wind. Wet conditions particularly favor its development. Gardeners can treat rose black spot with fungicides and by removing affected leaves. Good air circulation around plants also helps prevent infection. Finally, let's examine malaria. a disease caused by a protist parasite. The malaria protist has a life cycle that involves mosquitoes acting as vectors. Female mosquitoes spread the disease when they bite humans. When an infected mosquito bites a person, it injects the parasite into the bloodstream. The parasite then travels to the liver and begins multiplying. Malaria typically causes recurring episodes of fever, along with chills, sweating, and headaches. These fever cycles correspond to stages in the parasite's life cycle. These episodes can be very severe and, in some cases, fatal. Malaria is particularly dangerous for young children and pregnant women. The spread of malaria can be controlled by preventing mosquitoes from breeding. This involves eliminating standing water where mosquitoes lay their eggs. Using mosquito nets while sleeping also helps avoid being bitten, as does using insect repellent. In areas where malaria is common, people may take anti-malarial drugs to prevent infection. These medications help stop the parasite from developing in the body. Understanding these disease examples helps us appreciate how different pathogens affect living organisms in various ways. It also highlights the importance of prevention. Today, we're learning about the body's non-specific defense mechanisms, the barriers that stop pathogens entering in the first place, then examining what happens when pathogens breach these barriers and enter the body, and finally exploring how our immune system specifically targets and destroys different types of pathogens. Our body has evolved multiple layers of protection against disease. The first line of defense involves preventing pathogens from entering our bodies at all. The skin forms an effective physical barrier against pathogens. It provides a tough, waterproof covering over most of our body. When intact, it's very difficult for microorganisms to penetrate this barrier. Our skin doesn't just provide a physical barrier, it also produces antimicrobial substances that kill many pathogens on contact. This chemical defense adds another layer of protection. Inside our nose, we have tiny hair like structures and mucus that trap particles from the air we breathe. This prevents many potential pathogens from going deeper into our respiratory system. Similarly, the trachea and bronchi, the airways leading to our lungs, are lined with cilia, microscopic hair like projections. These tiny structures move in a coordinated way to waft mucus upwards to the back of the throat, where it can be swallowed. This mechanism is sometimes called the mucus escalator because it continuously moves trapped particles away from our lungs towards our digestive system. Speaking of our digestive system, the stomach produces hydrochloric acid, which creates a very low pH environment. This acidic condition kills many pathogens that might be present in our food or drink. But what happens if, despite all these defenses, pathogens still manage to enter our body? This is where our immune system comes into play. If pathogens breach our external defenses, our body activates internal defense mechanisms. White blood cells are the most important part of this immune response. White blood cells circulate in our bloodstream and can move into tissues throughout the body. They constantly patrol for foreign invaders, and when they detect pathogens, they respond in several ways. One method white blood cells use is called phagocytosis. In this process, certain white blood cells surround and engulf foreign organisms, then digest them using enzymes. This is like the white blood cells eating the pathogens. Another response involves the production of antibodies. When white blood cells encounter a foreign substance called an antigen, they produce specific proteins called antibodies. These antibodies lock onto the specific antigens on the pathogen's surface. This tagging helps other white blood cells identify and destroy the invader. It also prevents the pathogen from attaching to our body cells. If a person is infected with the same pathogen again, their white blood cells will rapidly produce the same antibodies. This quick response often prevents the disease from developing. We say the person has become immune to that specific pathogen. The third way white blood cells defend against pathogens is by producing antitoxins. These substances neutralize the toxins released by certain bacteria, preventing them from causing harm to our cells. Our defense systems work together to protect us from the countless pathogens we encounter daily. Without these mechanisms, even minor infections could become life threatening. Let's begin by going over exactly what we'll cover in this video. First, we'll examine how vaccination works to protect individuals from disease. Next, we'll look at how vaccinating a large portion of the population helps reduce the spread of pathogens. Finally, we'll discuss some of the benefits and limitations of vaccination programs. When you're infected with a pathogen, your immune system takes time to recognize the threat and mount a defense. During this learning period, you can become quite ill as the pathogen multiplies in your body. Vaccination works by preparing your immune system in advance without you having to suffer the actual disease. Vaccines typically contain dead or inactive forms of pathogens. These cannot cause the disease but still carry antigens, the molecular structures that trigger an immune response. When these weakened pathogens are introduced into your body, usually by injection, your white blood cells recognize them as foreign. This stimulates your immune system to produce antibodies that specifically target those antigens. For example, the MMR vaccine contains weak versions of the viruses which cause measles, mumps, and rubella. Each component triggers the production of specific antibodies against that virus. The clever part happens if you later encounter the real, active pathogen. Your immune system remembers the antigens from the vaccination and can rapidly produce the right antibodies to destroy the pathogen before it causes illness. This rapid response is what makes vaccination so effective. Your body doesn't need to go through the learning phase again. It's already prepared for that specific threat. Vaccination doesn't just protect individuals, it also helps protect entire communities through a concept called herd immunity or population immunity. When a large proportion of a population is vaccinated, pathogens cannot spread easily because they encounter fewer susceptible individuals. This creates a form of indirect protection for those who aren't vaccinated. This is particularly important for protecting vulnerable groups who cannot be vaccinated, such as very young babies, people with certain medical conditions, or those with severely weakened immune systems. Vaccines have had remarkable success in controlling once common diseases. In the UK, diseases like polio, measles, whooping cough, rubella, mumps, and tetanus have become rare thanks to widespread vaccination programs. Some diseases have been impacted even more dramatically. Smallpox, once a devastating global disease, has been completely eradicated worldwide through vaccination. Polio infections have fallen by 99% globally. Large outbreaks or epidemics can be prevented when enough people in a community are vaccinated. This breaks the chain of infection, as the pathogen cannot easily find new hosts to infect. However, if vaccination rates drop below certain thresholds, diseases can quickly resurge. When unvaccinated people are clustered together, pathogens can spread rapidly through these groups. It's important to note that while vaccines are extremely effective, they're not perfect. Some vaccines don't provide complete immunity for everyone who receives them. The protection may vary based on factors like age and overall health. Very rarely, people may experience side effects from vaccines. Most of these are minor and temporary, like soreness at the injection site or a mild fever. Serious reactions are extremely uncommon. In summary, vaccination works by safely exposing our immune system to pathogen antigens, allowing it to develop a rapid response if it encounters the real pathogen later. This not only protects individuals, but also reduces the spread of disease through populations. By understanding how vaccination works, we can appreciate why high vaccination rates are crucial for maintaining public health and preventing the resurgence of dangerous diseases. In this video, we'll cover three key topics: how painkillers manage symptoms without curing disease, how antibiotics fight bacterial infections, and why using these medications correctly is so important. When we're ill, medications can help in different ways. Some drugs treat the symptoms of illness, while others actually tackle the underlying cause. Painkillers, such as aspirin, are drugs that relieve pain, which is certainly helpful when you're feeling unwell. However, it's important to understand that painkillers don't actually tackle the cause of the disease or kill pathogens. Similarly, many over the counter medicines for colds and flu work by reducing symptoms like congestion, coughing, or fever. These cold remedies make you feel better, but don't actually fight the virus causing your cold. Antibiotics, on the other hand, work in a completely different way. Drugs like penicillin actively fight against bacterial infections by either killing bacteria or preventing them from reproducing. This is a crucial distinction. Antibiotics don't just make you feel better, they actually help eliminate the cause of the disease when that cause is bacterial. Different antibiotics are effective against different types of bacteria. This is why it's important to be treated with the right antibiotic for your specific infection. A key limitation of antibiotics is that they only work against bacteria. They have no effect on viruses that cause diseases like colds or flu. This is because viruses have a completely different structure and life cycle compared to bacteria. Viruses reproduce inside our body cells, which makes it very difficult to develop drugs that can destroy the virus without also harming our own cells. This is why many viral infections are treated by managing symptoms while our immune system fights the virus. The discovery and development of antibiotics has been one of the greatest medical advances of the 20th century. Before antibiotics, even minor bacterial infections could be life threatening. Since their introduction, antibiotics have greatly reduced the number of deaths from bacterial diseases. Infections that were once commonly fatal can now be effectively treated with a course of antibiotics. To summarize, painkillers and other symptom relieving medications help us feel better by treating the symptoms of disease. Let's begin by going over exactly what we'll cover in this video. First, we'll examine the historical origins of medicines and how traditional remedies influenced modern pharmaceutical development. Next, we'll investigate initial laboratory evaluation methods for potential new treatments. Then we'll look at how promising compounds are assessed in controlled human studies. Finally, we'll consider the importance of scientific verification before medications receive approval. Throughout history, the natural world has provided many therapeutic substances. Plant species synthesize various chemical compounds as defense mechanisms against insects and microorganisms. Remarkably, some of these natural substances can effectively treat human ailments. For centuries, people observed which plants seemed to offer medicinal benefits. These observations formed the foundation of traditional healing practices worldwide, many of which eventually led to contemporary pharmaceutical products. Consider aspirin, widely used for pain relief and fever reduction. This common medication originated from compounds identified in willow bark. Long before modern pharmaceutical science, people recognized willow's pain relieving properties. Similarly, the cardiac medication digitalis was developed from compounds present in the foxglove plant, which had historically been used for heart ailments. Microorganisms have also yielded important medicinal compounds. A noteworthy example comes from Alexander Fleming's laboratory work. While examining bacterial cultures, Fleming observed a curious phenomenon. A mold contamination had created a bacteria free zone in one of his culture dishes. This mold, identified as penicillium, was producing a substance that eliminated the surrounding bacteria. Fleming had discovered penicillin, inaugurating the antibiotic era in medicine. In contemporary times, pharmaceutical manufacturing occurs on an industrial scale. Although many medications still derive from natural substances, chemists frequently modify these compounds to enhance effectiveness and minimize unwanted effects. Before any medication becomes available to patients, it undergoes extensive evaluation through multiple testing phases. This rigorous assessment process ensures both safety and efficacy. Laboratory evaluation represents the first phase of this process. During this stage, researchers examine how compounds interact with human cells and tissues in controlled laboratory conditions. These experiments provide preliminary insights into potential therapeutic mechanisms. However, laboratory studies using isolated cells cannot fully predict how substances will behave in complete biological systems. For example, assessing cardiovascular medications requires evaluation in organisms with functional circulatory systems, not just isolated heart cells. Following successful laboratory studies, researchers conduct animal studies as part of preclinical evaluation. These investigations serve multiple purposes. They assess therapeutic effectiveness, identify potential toxicity issues, and help determine appropriate dosage ranges. UK legislation mandates testing proposed medications on two different mammalian species before human trials begin. This requirement generates significant debate. Some advocate for animal testing as a necessary safety measure, while others question its ethics and seek alternative methods. Compounds that successfully complete preclinical evaluation advance to human clinical trials, which proceed through several distinct phases. Initially, researchers administer experimental medications to healthy individuals. These early trials focus primarily on safety assessment rather than therapeutic benefit. Researchers begin with minimal concentrations of the compound, gradually increasing amounts if participants tolerate the substance well. When safety data from healthy individuals appears favorable, researchers then evaluate the medication in patients with the targeted condition. These studies determine optimal therapeutic concentrations, amounts that maximize beneficial effects while minimizing adverse reactions. To accurately assess effectiveness, researchers randomly assign participants to different treatment groups. Some receive the experimental medication, while others receive a placebo, an inactive substance appearing identical to the medication under investigation. This methodology allows researchers to distinguish actual medication effects from perceived improvements that occur simply because someone believes they're receiving treatment, the placebo effect. Many clinical studies employ blinding techniques to minimize bias. In blind studies, participants remain unaware whether they've received the actual medication or placebo. Double blind studies extend this principle further. Neither participants nor the immediate research team knows who received which substance until the study concludes. This approach prevents unconscious biases from influencing how researchers evaluate participants or how participants report their experiences. Before publication, research findings undergo peer evaluation. During this process, independent scientists scrutinize the methodology and conclusions to verify scientific validity and procedural integrity. This critical review mechanism helps prevent unsubstantiated claims about new medications and ensures that only thoroughly validated information reaches medical practitioners and patients. Developing new medications involves extensive resources, often requiring many years of research and significant financial investment. However, this comprehensive approach helps ensure that medications reaching patients offer genuine therapeutic benefits with acceptable safety profiles. Let's begin by going over exactly what we'll cover in this video. First, we'll explain what monoclonal antibodies are and how they differ from natural antibodies. Next, we'll examine the specific process scientists use to produce these antibodies. Finally, we'll explore their various applications in diagnosis, research, and treatment. Let's start by understanding what monoclonal antibodies actually are. Antibodies are normally produced by our immune system's B lymphocytes, a type of white blood cell. These Y-shaped proteins bind to specific foreign substances, helping our body identify and neutralize threats. When our immune system responds naturally, many different B lymphocytes produce slightly different antibodies. In contrast, monoclonal antibodies are identical copies that all target exactly the same binding site on one specific antigen. This remarkable specificity makes monoclonal antibodies incredibly useful tools. Because they only bind to one particular target, they can be used to identify or attack very specific substances or cells. Now, let's look at how scientists produce these specialized antibodies. The process begins by stimulating mouse lymphocytes to make a particular antibody. This is done by injecting a mouse with the specific antigen the scientists want to target. However, lymphocytes don't survive long in laboratory conditions. This presented a significant challenge when scientists first attempted to produce antibodies artificially. The breakthrough came when researchers discovered they could fuse these antibody producing lymphocytes with tumor cells to create what's called a hybridoma cell. This hybrid cell inherits two crucial characteristics: the ability to produce a specific antibody from the lymphocyte and the ability to multiply rapidly and indefinitely from the tumor cell. After creating these hybridoma cells, scientists isolate individual cells and allow them to multiply, creating clones. Each clone produces identical antibodies, all targeting the same antigen. The resulting population of identical cells all produce the same antibody. Scientists can collect these antibodies and purify them for various applications. Let's now explore some of the important uses of monoclonal antibodies. One common diagnostic application is pregnancy tests. These tests contain monoclonal antibodies that specifically bind to a hormone called HCG, which is only present in the urine of pregnant women. When a woman uses a pregnancy test, her urine moves along the test strip. If she's pregnant, the HCG in her urine binds to the antibodies, creating a visible line or symbol on the test. In medical laboratories, monoclonal antibodies are used to measure levels of hormones and other chemicals in blood samples. They can also detect specific pathogens, making them valuable tools for disease diagnosis. Research scientists use monoclonal antibodies to locate specific molecules within cells or tissues. By attaching a fluorescent dye to the antibodies, researchers can see exactly where their molecules of interest are located in a sample when viewed under a microscope. Perhaps one of the most significant applications is in treating disease, particularly cancer. Cancer cells often have specific markers on their surface that aren't found on normal cells. Monoclonal antibodies can be designed to recognize these unique markers. These therapeutic antibodies can work in several ways. They might be bound to a radioactive substance, allowing targeted delivery of radiation to cancer cells. Alternatively, they could carry a toxic drug that kills the cancer cells once the antibody attaches to them. Some monoclonal antibodies are linked to chemicals that stop cells from growing and dividing. In each case, the antibody delivers its payload specifically to cancer cells, minimizing damage to healthy tissue. This targeted approach differs significantly from traditional cancer treatments like standard chemotherapy and radiotherapy, which affect many body cells in addition to the cancer cells. Monoclonal antibodies precisely target specific cells, potentially reducing side effects. Despite their usefulness, monoclonal antibodies do have limitations. They can cause side effects that weren't initially expected. When first developed, scientists thought that because they target specific cells so precisely, they would have very few side effects. Additionally, the development of new monoclonal antibody treatments is costly and time consuming. This means they aren't as widely used in treatment as scientists originally hoped they would be. Understanding monoclonal antibodies helps us appreciate how scientific innovations can harness natural biological processes to create powerful tools for medicine and research. These laboratory created molecules continue to play an increasingly important role in diagnosing and treating a range of conditions. In our next episode, we'll explore how plants respond to pathogens and the various defense mechanisms they've evolved to protect themselves from. Let's start by outlining the main points we'll go over. First, we'll look at the common signs that indicate a plant might be diseased. Next, we'll examine the various types of pathogens that can infect plants. Then we'll explore the different methods used to identify specific plant diseases. Finally, we'll investigate the fascinating physical, chemical, and mechanical defense mechanisms plants use to resist infection. Plants, like all living organisms, are susceptible to diseases. Various pathogens can infect them, including viruses, bacteria, and fungi. We've already learned about tobacco mosaic virus and rose black spot in previous episodes. Plants can also be damaged by insects such as aphids, which feed on plant tissues and cause significant harm. When a plant is infected with a disease, it typically shows visible symptoms. Being able to recognize these signs is the first step in diagnosing plant problems. One common indicator of plant disease is stunted growth. Plants may be smaller than expected or grow more slowly than healthy specimens. This happens when pathogens interfere with the plant's ability to produce energy or transport nutrients. Spots on the leaves are another telltale sign of many plant diseases. These spots may vary in color, size, and pattern depending on the specific pathogen. For example, fungal infections often cause distinctive dark spots on leaves. Some plant diseases cause patches of decay or rot on leaves, stems, or fruits. These areas may appear soft, discolored, or water soaked. Plants suffering from certain diseases may develop malformed stems or leaves that appear twisted, curled, or otherwise abnormal compared to healthy plants. Discoloration is another important sign. Leaves may turn yellow, brown, or other unusual colors when the plant is infected. This happens when diseases affect the plant's ability to produce chlorophyll, or when pathogens damage plant tissues. If you're examining plants for potential insect infestations, careful observation often reveals the pests themselves on the plant. Looking at the undersides of leaves or at stem joints can help spot these unwanted visitors. Once you've detected signs of disease in a plant, the next step is identifying the specific problem. There are several methods for this. One approach is consulting gardening manuals or reliable gardening websites. These resources often include photographs and descriptions of common plant diseases, which can help match observed symptoms to specific pathogens. For more definitive identification, infected plant material can be taken to a laboratory where scientists have specialized equipment and expertise. They can examine samples under microscopes and perform tests to identify the pathogen. Modern testing kits are also available that use monoclonal antibodies to identify specific plant pathogens. These work similarly to the testing kits we discussed in our previous episode. The antibodies bind to particular pathogens, giving a visible result that confirms their presence. It's important to note that not all plant problems are caused by pathogens. Plants also require specific mineral ions from the soil, and deficiencies can sometimes produce symptoms that resemble disease. For example, plants need nitrate ions to make proteins necessary for growth. When nitrates are in short supply, plants often exhibit stunted growth, a symptom that could be mistaken for disease. Similarly, magnesium ions are essential for making chlorophyll, the green pigment needed for photosynthesis. Plants lacking sufficient magnesium may develop yellow leaves, chlorosis, and have reduced ability to photosynthesize. This yellowing might be confused with symptoms caused by certain pathogens. Now, let's explore how plants defend themselves against pathogens and pests. Unlike animals, plants can't run away from threats, so they've evolved sophisticated defense mechanisms. Plants have several physical defenses that act as barriers against pathogens. Most plant leaves and stems have a waxy cuticle that provides a waterproof barrier, making it difficult for pathogens to enter. Beneath this waxy layer, plant cells are surrounded by cell walls made from cellulose. These tough structures form another physical barrier against pathogens that manage to get past the waxy cuticle. Many plants, especially trees, have layers of dead cells around their stems. These form bark, which acts as an additional barrier to stop pathogens entering. If damaged, these outer layers can fall off, removing potential infection sites. Plants also employ chemical defenses. Some plants produce antibacterial chemicals that kill bacteria attempting to infect them. Examples include mint plants and witch hazel, which contain natural antimicrobial compounds. Other plants produce poisons that deter herbivores, organisms that eat plants. Tobacco plants, foxglove, and deadly nightshade all produce toxins that make them unpalatable or even dangerous to potential predators. In addition to physical and chemical defenses, plants have evolved mechanical adaptations to protect themselves. Some plants have adapted to grow thorns and hairs on their surfaces. These structures stop animals from touching and eating them by making the plant difficult or painful to consume. Other plants have evolved to drop their leaves or curl when touched. This response allows them to prevent being eaten by knocking insects off themselves or moving away from threats. Some plants go even further with mimicry as a defense strategy. For example, the bee orchid, Ophrys apifera, has evolved flowers that closely resemble female bees in appearance and even produce similar scents. This tricks male bees into attempting to mate with the flower, facilitating pollination in the process. Another fascinating example is the dead nettle plant, which closely resembles the stinging nettle but doesn't actually produce stinging hairs. By mimicking a harmful plant, it deters herbivores without having to invest energy in producing actual defensive structures. Some desert plants, like living stones, Isoaceae family, have evolved to look almost identical to the rocks and pebbles in their environment, making them nearly invisible to herbivores searching for vegetation to eat. While plants may seem passive, they have sophisticated systems for detecting threats and defending themselves against a hostile world. Today we're examining the basic photosynthesis reaction and its chemical equation, investigating the environmental factors that can limit its rate, looking at practical methods for measuring these rates, and finally exploring how plants utilize the glucose they produce. Photosynthesis is the process by which plants use energy from sunlight to convert carbon dioxide and water into glucose and oxygen. It's one of the most important biological processes on our planet. The word photosynthesis gives us clues about what's happening. Photo relates to light, and synthesis means putting together. So, photosynthesis is literally using light to build compounds. This process takes place in the chloroplasts of green plant cells. These specialized structures contain pigments like chlorophyll that absorb light. Chlorophyll specifically absorbs red and blue light while reflecting green light, which is why most plants appear green to our eyes. When light strikes these pigments, energy is transferred from the environment to the chloroplasts. This energy transfer powers the chemical reactions of photosynthesis. An important characteristic of photosynthesis is that it's an endothermic reaction. This means it requires energy input, in this case, from sunlight. The energy from light is transferred from the environment into the chemical bonds of glucose. We can represent photosynthesis with a word equation. Carbon dioxide plus water gives glucose plus oxygen. Or more formally, with a chemical equation, this equation shows us that six molecules of carbon dioxide combine with six molecules of water using light energy to produce one molecule of glucose and six molecules of oxygen. Now let's examine what affects how quickly this process occurs. The rate of photosynthesis can be limited by several environmental factors. The first limiting factor is light intensity. Since light provides the energy for photosynthesis, the rate typically increases as light intensity increases. However, this relationship isn't unlimited. As the graph shows, the rate increases steadily with light intensity only up to a certain point. Beyond this point, increasing light intensity no longer increases the rate of photosynthesis. This happens because another factor has become limiting. When light is abundant, either temperature or carbon dioxide concentration typically becomes the next limiting factor. Carbon dioxide concentration is our second limiting factor. CO2 is one of the raw materials needed for photosynthesis. Similar to light intensity, as CO2 levels increase, the rate of photosynthesis increases, but only up to a point. The graph flattens out when another factor becomes limiting. Temperature is the third major limiting factor. Photosynthesis involves enzyme controlled reactions, and these enzymes are affected by temperature. At low temperatures, the enzymes work more slowly, limiting the rate of photosynthesis. As temperature increases, the rate of photosynthesis increases as enzymes work more efficiently. However, if temperatures get too high, for example, around 45 degrees Celsius, the enzymes begin to denature or become damaged. This causes a rapid decline in the photosynthesis rate. The amount of chlorophyll can also limit photosynthesis. If a plant has been affected by disease or lacks nutrients, like magnesium, its chloroplasts may be damaged. Magnesium is needed to make chlorophyll. Without enough chlorophyll, the plant's ability to absorb light decreases, reducing photosynthesis rates. These limiting factors don't work in isolation. They interact with each other. At any given moment, one factor will be the limiting factor. This is the factor that, if changed, would have the greatest effect on the rate of photosynthesis. For example, on a bright summer day, carbon dioxide might be the limiting factor for a plant. Adding more CO2 would increase photosynthesis rates, but adding more light wouldn't help because light is already abundant. We can see these interactions in graphs that show the effect of multiple factors. For instance, a graph might show photosynthesis rates at different light intensities for two different temperatures. At the lower temperature, such as 15 degrees Celsius, the line levels off at a lower point than at the higher temperature, such as 25 degrees Celsius. This shows that temperature becomes limiting once light is abundant. Similarly, graphs showing the effects of light intensity and CO2 concentration demonstrate that at higher CO2 levels, the plant can use more light before another factor becomes limiting. Scientists measure photosynthesis rates in various ways. One common method uses aquatic plants like Canadian pondweed. As the plant photosynthesizes, it produces oxygen, which forms bubbles. The rate at which bubbles are produced corresponds to the rate of photosynthesis. In a typical experiment, a light source is placed at a specific distance from the pondweed. The plant is allowed to photosynthesize for a set time period, and the oxygen produced is collected in a capillary tube. The length of the gas bubble in the tube is proportional to the volume of oxygen produced. This setup allows scientists to investigate how different factors affect photosynthesis. They might vary light intensity by changing the distance between the light source and plant. When using light as a variable, it's important to understand the inverse square law. This states that light intensity decreases in proportion to the square of the distance. For example, if you double the distance between the light source and plant, the light intensity becomes 4 times smaller. If you triple the distance, intensity becomes 9 times smaller. Now, let's consider what plants do with the glucose they produce through photosynthesis. Plants use glucose in 5 main ways. First, glucose provides energy through respiration. Just like animals, plants need energy to power their cellular processes. They obtain this energy by breaking down glucose during respiration. Second, plants convert excess glucose into starch for storage. Starch is insoluble, making it better for storage than glucose. Glucose is soluble and would draw in water, causing cells to swell up. Third, glucose can be converted into cellulose, which is used to build strong plant cell walls. These walls provide structure and support for the plant. Fourth, plants can combine glucose with nitrate ions absorbed from the soil to make amino acids. These amino acids are then assembled into proteins. Proteins serve many vital functions, including acting as enzymes that control chemical reactions in the plant. Finally, glucose can be stored as fats and oils, particularly in seeds. These provide a concentrated energy source for future germination and growth. Understanding photosynthesis and its limiting factors has important practical applications. In agriculture, farmers and gardeners create controlled environments like greenhouses to optimize conditions. Greenhouses help trap heat, maintaining optimal temperatures even when outside conditions are unfavorable. They can be equipped with heaters in winter or ventilation and shade in summer. Commercial growers often supplement natural light with artificial lighting. This ensures plants receive adequate light even on cloudy days or during shorter winter days. Carbon dioxide levels can be increased by using paraffin heaters or by adding CO2 directly. Enclosed greenhouse environments also make it easier to protect plants from pests and diseases. By manipulating these limiting factors, farmers can create ideal conditions for plant growth. This leads to increased yields and more efficient crop production. Before we get into the details, here's what's coming up. First, we'll clarify what respiration really is and how it differs from breathing. Next, we'll examine the two types of respiration, aerobic and anaerobic. Then we'll look at how our bodies respond during exercise when energy demands increase. Finally, we'll explore the concept of oxygen debt and how our bodies recover after intense exercise. Respiration is the process of transferring energy from the breakdown of glucose. It happens continuously in every living cell of your body. It's important to understand that respiration is not the same as breathing. Breathing, or ventilation, is simply moving air in and out of your lungs. Respiration, on the other hand, involves many chemical reactions that transfer the energy cells need for all living processes. All living things respire, not just animals, but plants, fungi, and even single celled organisms. It's how they transfer energy from their food to their cells. Respiration is an exothermic process. This means it releases energy in the form of heat as well as usable energy for the cell. There are two main types of respiration, aerobic and anaerobic. The key difference is whether oxygen is used in the process. Aerobic respiration uses oxygen. It's the most efficient way to transfer energy from glucose. The word equation for aerobic respiration is glucose plus oxygen equals carbon dioxide plus water. There is also a chemical form of this equation. Aerobic respiration happens all the time in plants and animals. Most of the reactions take place inside specialized structures called mitochondria. This process releases a large amount of energy from each glucose molecule because the oxidation of glucose is complete when oxygen is available. Anaerobic respiration occurs when there isn't enough oxygen available. The word anaerobic literally means without oxygen. In human muscle cells, anaerobic respiration produces lactic acid. This incomplete breakdown of glucose releases much less energy than aerobic respiration. This is because glucose isn't fully oxidized in anaerobic conditions. Plants and yeast cells can also respire without oxygen, but they produce different end products. Their anaerobic respiration creates ethanol and carbon dioxide instead of lactic acid. This process in yeast cells is called fermentation. It has important economic applications in bread making and producing alcoholic drinks. In bread making, it's the carbon dioxide from fermentation that makes bread rise. In beer and wine making, it's the fermentation process that produces alcohol. Now let's look at how our bodies respond to exercise when energy demands increase dramatically. When you exercise, your muscles need more energy from respiration to contract more frequently than normal. This increased need for energy means your cells need more oxygen and must remove carbon dioxide more quickly. To meet this demand, several things happen in your body. Your breathing rate and breath volume increase. This gets more oxygen into your blood and removes carbon dioxide more efficiently. Your heart rate increases to pump this oxygenated blood around your body faster. During moderate exercise, these adaptations are usually sufficient to supply your muscles with the oxygen they need for aerobic respiration. However, during vigorous exercise like sprinting, your body cannot supply oxygen to your muscles quickly enough. When this happens, your muscles start respiring anaerobically as well as aerobically. This produces lactic acid, which builds up in the muscles and can cause pain and fatigue. Long periods of exercise can cause muscle fatigue. The muscles get tired and stop contracting efficiently. After intense exercise involving anaerobic respiration, you'll have what's called an oxygen debt. This is the amount of extra oxygen your body needs to recover after exercise. This oxygen is used to react with the lactic acid that has built up and remove it from the cells. In other words, you need to repay the oxygen that you didn't get while you were exercising intensely. This is why you continue breathing hard for a while after you stop exercising. Your pulse and breathing rate stay high while there are high levels of lactic acid and carbon dioxide in your blood. Your body also has another way of coping with lactic acid. Blood that enters your muscles transports the lactic acid to the liver. In the liver, the lactic acid is converted back into glucose. Understanding respiration helps us appreciate how our bodies produce the energy needed for everything from basic cellular functions to intense physical activity. In our next episode, we'll explore metabolism. Today, we're defining metabolism and its relationship to cellular activities, looking at how enzymes control metabolic reactions, examining synthesis reactions where cells build large molecules, and finally exploring the breakdown reactions that release energy. Metabolism encompasses all the chemical reactions that happen in an organism. Every cell in your body is constantly carrying out thousands of chemical reactions. These reactions allow your body to grow, reproduce, maintain its structures, and respond to its environment. All of these reactions are controlled by enzymes. Enzymes are biological catalysts that speed up chemical reactions without being used up themselves. Many of these reactions are linked together to form sequences or pathways. The product of one reaction becomes the reactant for the next. Metabolism includes two main types of reactions, those that build up complex molecules and those that break down molecules. Let's look first at how cells build larger molecules from smaller ones. These synthesis reactions are essential for growth and repair. One important example is the formation of carbohydrates. Lots of small glucose molecules can be joined together to form larger molecules. In plant cells, glucose is converted into starch for energy storage and cellulose for cell walls. In animal cells, glucose molecules are linked to form glycogen, which is stored in the liver and muscles. Lipid molecules are another example. They're synthesized from two types of building blocks, glycerol and three fatty acids. These lipids are vital for cell membranes and energy storage. Proteins are formed when glucose is combined with nitrate ions to make amino acids. These amino acids are then linked together to form proteins. Proteins serve countless functions in your body, from structural components to enzymes that control reactions. Just as important as building things up is breaking them down. Your body also needs to break larger molecules into smaller ones. The most significant breakdown reaction is respiration. As we saw in our previous episode, respiration breaks down glucose to release energy. This energy powers all the other reactions in the body that make molecules. Another important breakdown process happens with excess proteins. These are broken down in a reaction to produce urea. The urea is then excreted in urine, removing nitrogen waste from the body. Metabolism is a careful balance between these building up and breaking down processes. Both are essential for life. Together, they maintain the delicate balance that keeps you alive and functioning.